Hi everyone, this is Miss Moriarty. I'm here to discuss with us our topic 8.3, 8.7, and 8.8 lecture video. As you watch the video, make sure you take down notes. Whenever you are done, please mark this video as complete, and please attach a picture of your completed notes to this assignment. All right, so here we go. Topic 8.3, everybody, is about endocrine disruptors. These are chemicals that are going to interfere with our endocrine system. Now, our endocrine system is responsible for the regulation of all the different types of hormones that we might find within our bodies. Why endocrine disruptors are so dangerous is because they can bind to cellular receptors meant for hormones. So they can kind of act like hormones that we find in our bodies, and they can either block the hormone from being received or even amplify its effect. Now, as a result, endocrine disruptors typically lead to things such as birth defects, developmental disorders, and even gender imbalances, especially in fish or even in other species. And here in the image to the right here, these are just some common examples of endocrine disruptors and where you might find them. Here's just a little more complicated list of your impacts of endocrine disruptors. The typical sources can come from things such as agricultural runoff. It could come from industrial effluents, municipal effluents, meaning from homes that are cities. So mainly our impacts or our sources of endocrine disruptors can typically come from anything that's around us. And here's just a simple list. I recommend becoming familiar with this list. This is the Dirty Dozen list of endocrine disruptors. So I would be familiar with each of these. You'll see that most often in FRQs, I've seen the mentioning of atrazine. I've seen mercury and lead, as well as arsenic. These are all very common endocrine disruptors that are mentioned in FRQs. Now, moving on to 8.7. Now, 8.7, this is all about persistent organic pollutants. So let's kind of break this word down here. So POPs, for short, are substances that don't easily break down in the environment, hence why we call them persistent, but they're also synthetic and carbon-based, hence why we say organic. Perfect examples of those are two substances known as DDT and PCBs. POPs are very toxic to organisms because they're actually soluble in the fat tissues of organisms. And so what that does is it allows that chemical to then accumulate in the organism's fatty tissue, hence bioaccumulation. POPs can also travel over very long distances via wind and water before even being redeposited in different ecosystems. Back in 2001, there was actually even a group of 127 nations or so that gathered in Stockholm, Sweden, that actually reached an agreement and found that they need to restrict the use of some of these chemicals. And so there were actually 12 chemicals that were banned or phased out. And those are actually the original Dirty Dozen. These all 12 chemicals here are considered to be POPs. Each one is either not used anymore, is greatly reduced around the globe, because they found that these chemicals were in top predatory organisms, and it was causing issues amongst those types of organisms, especially even keystone species. You can find that there's a pretty pattern here. A lot of the POPs tend to come from things such as insecticides, some fire retardants, as well as industrial products. Now let's get into bioaccumulation and biomagnification. I'm sure you may have heard these two words before, and I know our lab is on both of these topics here. But the difference between the two, in bioaccumulation, this is where we see the selective absorption and concentration of elements or compounds by cells in that living organism. And they're usually going to be found in they're going to be found by fat-soluble compounds. But in biomagnification, this is the phenomenon that tells us that we're going to see an increase in concentration of those substances per unit of body tissue that occurs in then higher trophic levels of a food chain or in a food web. Now, we know back from Unit 1, when we talked about food chains and food webs and the energy pyramid, we know things such as primary consumers, especially in aquatic ecosystems, zooplankton, bottom-feeding fish, insects. They can take in POPs simply just by eating producers such as algae, and that causes bioaccumulation of the POPs in their tissues. Those secondary consumers then eat those primary consumers and take the POPs into their own tissues. And because of that 10% rule, we know that organisms at each successive trophic level need to eat more and more biomass in order to receive enough energy to survive, leading to higher and higher POPs levels over their lifetimes. And so species that are quite large, such as salmon, dolphins, whales, they have tremendously high levels of POPs or even methylmercury levels in their systems. So let's specifically look at the biomagnification of mercury. Now, we know mercury tends to come from burning fossil fuels, right? A coal-fired power plant is going to unfortunately emit lots of mercury. A natural source could be a volcano. Now, that mercury can be carried by wind, deposited in water, and bacteria will then convert it into then an even more toxic chemical known as methylmercury. That is then taken in by phytoplankton and it's biomagnified at much higher trophic levels. Once you reach that high level, we're going to find lots of that then methylmercury in those top predators. Once it reaches that very high level and that accumulation has now tremendously occurred, that becomes a neurotoxicant. So it has the potential to damage the central nervous system of those then higher predatory animals. Even as humans, when we consume fish, humans can be exposed to methylmercury as well, especially since we are quite high on the food chain as well. And so that methylmercury and POPs can actually come from eating things such as tuna and salmon. So that's usually why they recommend only eating so much tuna, because it does have the potential to damage our human nervous system as even developing fetuses. Now, what about the biomagnification of DDT? Well, DDT, guys, that was a pesticide that was used. It was banned in many developed nations, but it still persists in sediments and many other bodies of water. It's taken in by bottom feeders, zooplankton, and obviously biomagnified at higher trophic levels. It reaches that high level of top predator, especially things such as birds, eagles, osprey. It was actually the accumulation and magnification of DDT that led to the bald eagles becoming endangered. What it did was it caused thinning of their eggshells. And so lots of those bald eagles were then unable to produce offspring, leading to then a reduction in their population size. But now, since the passage of the Endangered Species Act, the bald eagles are, I believe, no longer endangered. Now, some other laws that we need to understand that helps to protect our waterways. These two we also discussed in the very beginning of the year as well. And you might often see these embedded in FRQs about water pollution. One being the Clean Water Act that helps to support the protection and propagation of fish, shellfish, wildlife, recreation. This is issued water quality standards that define acceptable limits of various pollutants in all U.S. waterways. This is also where we help to mitigate and protect wetlands and mangroves. Remember, that's kind of falls under the Clean Water Act umbrella there. While the Safe Drinking Water Act, that actually sets the national standards then for drinking water. So it is responsible for establishing what's called a maximum contaminant level for at least 77 different elements or substances in both surface and groundwater. And just so you can kind of see what I mean by that, right here in this table, the MCL for a variety of contaminants in drinking water is determined by the EPA in parts per billion. So here you can see a safe level of drinking water might have some arsenic in it, some mercury, benzene, atrazine. All of these contaminants, unfortunately, are in our waterways, but they have determined that these are safe levels. We can also see in the next table the current leading cause and sources of impaired waterways in the US. So we see in streams and rivers, we often see lots of bacterial pathogens, habitat alteration, oxygen depletion, usually comes from things such as agriculture, the diversion of water, the construction of a dam. Lakes, ponds, and reservoirs, we see lots of mercury, PCBs, nutrient pollution that comes from agriculture, atmospheric deposition. Bays, estuaries can usually be bacterial pathogens, once again, oxygen depletion, as well as mercury that can come from municipal discharge, such as sewage, or even atmospheric deposition. The then practice FRQ. So if at this point you want to just pause the video and see if you can answer your practice FRQ, explain why there are greater health risks associated with eating large predatory fish, such as tuna, sea bass, than from eating small non-predatory fish. Okay, at this point, you should have paused the video and see if you can answer the FRQ. And here are the accepted answers here from College Board. So your explanation should have been based on your idea or phenomenon known as bioaccumulation and biomagnification. So you could have mentioned things such as mercury transfers to higher trophic levels. Concentration of mercury is going to increase in those higher trophic levels. Here would be an example, right? Over the course of a lifetime, older fish have had more time to accumulate those significant amounts of mercury, regardless of their trophic level, is also another type of point that you could have mentioned. All right, guys, that's it for this video. You want to make sure that you have watched the video, take your notes, and please take a picture of your notes and attach them to this assignment.